so much for coming out on this lovely afternoon. I am Ruth Huffbauer, and I'm the director of the Ecology program here at CSU. And I am today also the host of our distinguished speaker. And so I get to introduce Chris. A few announcements first. We're going to go back to Avos for our spring picnic and graduation party, so look for emails about that. That should be fun. The old guard will remember we used to have spring parties there at Avos a lot. Good place to go. That's all I'm thinking of at the moment. So it is my great pleasure to introduce Chris Funk. He is an evolutionary biologist, does ecological, evolutionary ecology research as well, but I have admired him for a long time for his just really amazing evolutionary biology with a clear link to conservation and application. And his publication record is phenomenal. Yet he also really engages in that conservation side. So he is he consults with the U.S. Fish and Wildlife Service, for example. He is on various international committees focused on conservation and biodiversity. And he literally is one of the people who wrote the book on conservation and the genomics of populations. So we all have a lot to learn from him. A little bit about just background. His Ph.D. was from University of Montana, then he postdoced in University of Texas at Austin, and did another postdoc with the USGS in Corvallis, and a lot of his research is focused on amphibians in the Oregon area and at my house. So we're having a reception at my house if you haven't seen the email. So I want to celebrate those kind of Oregon roots. So a few other things about Chris. He's from here, right? This is the Distinguished Ecologist Seminar series where we're typically bringing in folks from other places. And when the executive committee looked at the nominations last year, they said, well, if Chris weren't at CSU, we would invite him to come give a talk. So we should just have him talk in the spring. And that is so true. Looking at his the list of talks that he's given, he's the student invited speaker at McGill, at Utah State, at far too many for me to remember. And that is really an honor to be chosen by the students of a program to go. AAAS Fellow, does tons of work with SOGIS, has an amazing grant record with 10 major NSF grants, which is impressive. But most of all, I really, I think you'll enjoy Chris's science. It's important and powerful evolutionary stuff. With that, take it away.
Cool. Thanks, Ruth, for that overly generous introduction. So it's a real pleasure and honor today to be today's GDPE Distinguished Ecologist. In my opinion, GDPE is one of the best things about CSU, and it's one of the main things that's actually kept me here at CSU. But ironically, about a year ago, GDPE was one of the main reasons that I almost left CSU. And that's because when Ruth invited me to be a GDPE Distinguished Ecologist, she emailed me a very clear message saying, on behalf of CSU, GDPE, I'm honored to invite you to attend our university as one of our visiting distinguished ecologists for spring 2026. So Ruth made it absolutely clear that it was time for me to go if I wanted to be a GDPE distinguished ecologist. And so I took this requirement very seriously, as many people in this room know. I started looking for jobs elsewhere, got some interviews and offers, and I almost ended up taking them. But then, sort of at the last minute, Ruth said that she would allow me to still be a distinguished ecologist if I stayed at CSU, so the best of both worlds. As long as I did my best to try to at least seem distinguished and not embarrass GDPE or CSU too much. But hopefully I've done a good job of embarrassing Ruth. All right, so today I'm going to tell you a frog tale about a tailed frog. Or if you prefer a more academic sounding title, which some of us are more comfortable in the academic realm, why an evolutionary perspective matters for predicting sensitivity to increasing temperatures. I always like to learn about people's stories of why on earth they decided to become a biologist. So I thought I'd start off with a little bit about my story of how I became a biologist focused on biodiversity conservation. Next, I'll provide a little bit of context for the research I'll talk about today, provide a little theory on vulnerability to climate change. And then I'll focus on the empirical study on our research using tailed frogs as a window into trying to understand vulnerability to climate change, focusing on Pacific Northwest streams. And some of the results I'll be presenting today are pretty brand new, so I'd like your feedback on them. And lastly, I'll finish with some concluding thoughts about what we're learning about vulnerability to climate change, which is obviously an extremely important topic. It's a topic that everybody and their dog is thinking about now, but it's also an incredibly daunting topic. All right, so first we'll talk about my story. So I feel really fortunate to have grown up in the beautiful and ecologically diverse state of Oregon. So most of the time when people think about Oregon, they think about the wet western part of the state. But actually over half of Oregon is high desert. And so you can go from the coast or the Western Cascade ranges where you get about 160 inches of rain a year. So we're talking like over 4 meters of rain, to the high desert in about 20 kilometers distance, and you get down to about 20 centimeters of rain. So a really fascinating place to grow up and become interested in biodiversity. And I grew up on this hill called Cooper Mountain in the northern Willamette Valley, which of course now has a winery on it, which was played a huge role in why I became a biologist focused on biodiversity. So, like a lot of people probably in this audience who have decided to go into biology, I've loved being outside as long as I can remember. So as a kid, this is back in the day when I could just kind of roam the hill and the forest and explore streams. And if I wanted dinner, I should probably show up around 6 p.m., but my mom wasn't like looking for me. I didn't have a cell phone. And so I spent lots of time outside as a kid exploring Cooper Mountain. And then when I became a young adult, I continued to explore the outdoors any way I could, backpacking, rock climbing. I was a wildlands firefighter for some summers. And my favorite activity was whitewater kayaking because streams were always my favorite environment. But then something happened that turned a budding young biologist into a budding young conservation biologist, which was the clear cutting of my childhood forests. So I took these photos when I came home from college one summer, and this really pissed me off. I'm a young person, and it broke my heart, but it also inspired me to try to make a difference. And this is really what set me on the path to becoming a conservation biologist. And I've heard really similar stories from other people in the field, like Michael Soule, who's considered sort of the founding father of conservation biology, who decided to essentially invent the field of conservation biology when he witnessed his childhood homes, the Arroyos outside of San Diego, being destroyed, which was exactly what was happening to my precious Cooper Mountain. And although I didn't know it at the time, what I was witnessing is part of a larger phenomenon that has been termed the Great Acceleration. And I think these images do a really good job of capturing the unparalleled rate of change in human population size. Got that here. And just look at the axis. That's starting in 750. So if go out like humanity starts over there. And all this has happened very recently. Per capita consumption, McDonald's restaurants, go on and on. And our planet has never endured anything quite like this in its 4.5 billion year history. And I think it's important to realize that we are not in normal times. Of course, one of the most devastating aspects of this great acceleration is habitat loss and fragmentation, represented here by slash and burn agriculture. And of course, the one we're thinking more and more about is climate change, that increasingly threatens humans and biodiversity alike. So mean temperature has already increased by 1 degree Celsius above what it was in the Industrial Revolution, and it's expected to increase a lot more even under the most optimistic scenarios. Oh, there's my son sending me a text. And unfortunately, it looks like we're headed towards, oh, this is the one that takes a long time to go because it's a huge image. This is not going because I touched my text. Huh. There we go. But unfortunately, it looks like we're headed towards one of the more pessimistic scenarios with repeated records of temperature. And I think this is an image that was in the New York Times, I think just a couple of years ago. And this is really striking because it shows this is change over seasons, and this is what it's looking like in 2023. Now, I love world records, but this is not a world record you want to break. And just seeing these figures of increases in temperature can feel very abstract and impersonal, but climate change affects all of our lives more and more each year, and more directly, as we know all too well in the US West, where we've had epically grim winter snowpack in 2025, 2026, also known as the winter that never was. And here's where we're at in the South Platte snowpack this year. So this is almost certainly be followed by one of the most epically grim fire years we've ever witnessed. So one of the biggest scientific questions of the 21st century, not just in biology, but biggest scientific questions, period, is how will biodiversity respond to this incredibly rapid environmental change? And what can we do to maximize the resilience of biodiversity to this rapid change? So this leads me to the goal of my research program, which is to strive to understand the evolutionary ecological mechanisms that generate and maintain biodiversity. Obviously, this is a huge question. We're only going to take a bite out of it. And at the same time, I strongly believe it's our responsibility as ecologists and evolutionary biologists to communicate our research and work with managers and policymakers who are trying to make the best decisions possible that affect biodiversity and sustainability. And so my favorite projects are those that simultaneously test basic theory and that also directly inform conservation. So my research is really integrative. I combine field research, experiments, population genomics, and computational approaches. And usually the responsibility of our group is to lead the population genomics component of projects. But my heart will always be in the field, which is where I got my start, romping around the forests and streams of Cooper Mountain. We also work on a huge variety of organisms, mainly things with backbones, but sometimes things with exoskeletons, represented by some of the critters we've worked on here. We also work a lot of different locations across the world. I realize when I show this that this is totally outdated. There's a lot more red pens now, including in the Eastern Hemisphere. And it's clear from the Funk Lab logo, my favorite ecosystems are mountain streams and other freshwater habitats. So there's a lot of awesome things about our jobs. I think I have the best job in the world. But I would say my favorite part is mentoring the next generation of biologists. So this slide shows some of the graduate students, postdocs, research scientists, lab managers, and undergrads that I've had the pleasure of working with since I got to CSU almost 18 years ago. And We make a special effort to recruit diverse students and postdocs because science, which after all is the pursuit of truth using experiments, observation, and logic, that belongs to everybody. And I also strongly believe that having diverse people in your group is essential to address these hairy problems involved with biodiversity conservation. In terms of research topics, I like to think of my research program as a tree. The overarching motivation behind my research program is to understand the evolutionary and ecological mechanisms that generate and maintain biodiversity and to form conservation of biodiversity. Then there are 4 main branches of investigation in my lab. The first branch is conservation and population genomics. Another branch is climate change, vulnerability, sensitivity, and resilience. Another focuses on the interaction between adaptive divergence and gene flow. And then another branch is on tropical ecology and evolution conservation, because although a lot of my research focuses in the western US, also a lot of it is in South America, particularly in northwestern tropical South America. And then these leagues can be thought of as specific projects. So in the area of conservation genomics, Ruth was talking about, I co-authored this text called Conservation and the Genomics of Populations with Fred Allendorf, who's the one who originally wrote the first and second edition. And this is the first comprehensive book on harnessing genomics for addressing a variety of questions in conservation. Fred has passed the baton to me to lead the 4th edition, at which point I said, I don't know if this is an honor or a curse, and he just said yes. And when we spent 3 years obsessively working on this book, it's a pretty awesome feeling to finally hold it for the first time. And on a brisk spring day, wearing a flannel shirt, which is probably the reason that my entire lab decided to do the same thing last week on April 1st, which it took me 2 days to notice on their profile pictures, demonstrating my excellent powers of observation as a scientist. Nice one, Funklings. So another project focuses on testing the mechanisms causing speciation in megadiverse tropical countries like Ecuador and Colombia, where they have, 10 times more frog species than we do in all of the US. A lot of work on microgeographic adaptation, interactions of adaptation and gene flow. And another project focuses on vulnerability to climate change and the tailed frog. And that's what I'm going to focus on today. All right, so now you know a little bit about my background, what motivates me, and the different branches of my research program. I'd like to provide some context for the research I'll talk about today by presenting some theory on vulnerability to climate change. So biodiversity is predicted to experience some severe effects from increasing temperatures and from other effects of climate change. So this is a figure taken from a paper that Mark Irvin published several years ago in Science, and it shows percent extinction in communities as a function of projected increases in pre-industrial temperatures. And so if we're able to meet the policy target of less than 2 degrees Celsius, then we project, based on this, that about 5%, the extinction rates will increase about 5%. But we're more like over here, so we're looking at more like a 10% increase in extinction probability. And this may not seem like a lot. You may be willing to live with a 10% increase in extinction probability. But remember that this doesn't include all the other stressors that biodiversity faces of habitat loss and fragmentation and pollution and invasive species and overexploitation, et cetera. So one thing you'll notice from this is that, there's an increase, there's a positive line here. So the higher temperature gets, the greater probability of extinction, more of the community is expected to be lost. But there's a ton of variation around this curve, right? And so that variation is due to real variation among communities and species and their probability of going extinct in response to climate change, but also due with a lot of uncertainty in these predictions. So one way we can help move solutions forward as scientists is to do a better job of predicting vulnerability. But what is vulnerability? So vulnerability, there's different models, but the one I'm going to use is vulnerability is a product of sensitivity times exposure minus adaptive capacity. All right, let's break down each of these terms. So vulnerability is the probability of extinction in response to climate change. So that's essentially the y-axis in that Mark Urban figure. Sensitivity is the response of a population or species per unit change in temperature. And oftentimes sensitivity is conflated with vulnerability, but these are different because vulnerability is also a product of exposure. And this is just the amount of change in temperature or change in precipitation. And so vulnerability is the product of sensitivity and exposure, but it's also moderated by adaptive capacity. And adaptive capacity is the ability of a system to adjust to climate change from dispersal, from plasticity, or from evolution. So the higher adaptive capacity is, the lower your ultimate vulnerability is going to be. So if we want to do a better job of predicting vulnerability, we have to do a better job of understanding sensitivity, exposure, and adaptive capacity. It's pretty well known that genomics and evolution are relevant for understanding dispersal or gene flow and also adaptive potential. But today I also want to argue that evolutionary theory and genomic tools is also really important for understanding variation and sensitivity of populations and species to climate change. So for now, just focusing on sensitivity, to do a better job of understanding how sensitive populations of species are, we have to focus on what I'll call sensitivity traits. So some examples of traits that determine sensitivity to climate change are things like thermal tolerance is an obvious one, drought and flood tolerance, and if you're a marine organism, particularly one with shells, then tolerance of acidification is really important. But obviously climate change is not the only dimension of environmental stressors. So you can also imagine sensitivity traits related to disease resistance or competitive ability against invasives. So this is just a small subsample of sensitivity traits, but the key point is that traits are key in order to predict vulnerability to climate change and other stressors. So going one level deeper, we can ask what determines variation and sensitivity traits among and within species. So today I'll talk a lot about within species or among population variation and sensitivity. So my colleagues and I came up with a simplified conceptual framework to help us think about this. And this framework starts off with landscape and environmental heterogeneity that organisms and populations experience in their evolutionary history, which in turn affects microevolutionary processes like selection and genetic drift and gene flow. And these processes mold the amount and spatial distribution of genetic variation. And this genetic variation in combination with environmental effects determines the distribution of phenotypic variation. And here I'm talking specifically about sensitivity traits. And finally, variation in these sensitivity traits in combination with exposure determined by environmental variation ultimately determines patterns of variation and vulnerability. And so the general idea here is that environmental heterogeneity will mold the distribution of sensitivity traits, which in turn affects spatial patterns of variation and vulnerability. So there's nothing groundbreaking about this framework. It's basically the way that most evolutionary biologists think about the world. But what's different is the focus on sensitivity traits and vulnerability. So today I want to make the case that understanding local adaptation and sensitivity traits is really important for determining which populations are predicted to be most vulnerable to increases in temperature or other dimensions of climate change. And this is super important because this is the information that managers and conservation practitioners need to make decisions. Just knowing which species are most vulnerable is information that's really difficult to use on a management scale. They need to know if certain populations are particularly vulnerable or relatively resilient for making decisions about what they're going to do on the ground. All right, so this first panel shows a common paradigm in a lot of vulnerability assessments in which there's no variation among populations across environmental gradient and sensitivity. So let me break it down. The blue curve shows temperature as a function of elevation, but this could be any kind of temperature gradient. The dashed blue line shows current CT max. So CT max is the maximum temperature an organism can tolerate before it essentially loses bodily function. And then this orange curve shows expected increases in temperature with climate change. And so under this model, in this common paradigm, the populations that are most likely to be vulnerable first are those low elevation populations, because they're going to be the first to experience temperatures above their CT max. But imagine there's evolution and there's local adaptation. If that's the case, then you might expect CT max to change in parallel with historical temperatures that populations have experienced in their evolutionary history. And if that's the case, then all populations across that elevational gradient might be equally vulnerable or equally resilient. Or you can imagine a scenario in which low elevation populations evolve a higher CT max quicker than high elevation populations because they have more genetic variation. And that's a pattern that's been seen in lots of different organisms. And if that's the case, then you might end up in a case where the high elevation populations are going to be the most vulnerable to increases in climate change. So you can keep on making hypotheses until you blue in the face, but the point here is that understanding variation in sensitivity and local adaptation and sensitivity is really important for predicting which populations are going to be most sensitive to increases in temperature. All right, so what I'd like to focus on now is our research using tailed frogs as a window into understanding vulnerability to climate change in Pacific Northwest streams that look like this. And I was telling someone today, one of the students I met with, that I largely choose my research projects based on where I get to work. And so I find a place like this, I'm like, okay, what lives here and what's some cool questions that somebody's willing to fund? And so this project was mainly funded by an NSF program called Rules of Life. And the title was Predicting Vulnerability to Climate Change by Linking Environmental Heterogeneity to Genetic and Phenotypic Variation. And one of my favorite things about this project is bringing together a lot of my favorite scientists. On our genomic team, we had Brenna Forrester, my former postdoc, who's now endangered species biologist with the U.S. Fish and Wildlife Service. Our physiology team was led by my former graduate student, Dr. Amanda Chikina, who's now a postdoc at the University of Guelph in Canada. Also, Alicia Shaw, who was co-advised by Cameron Gallenbohr and myself, and Cameron Gallenbohr, who is now at Norwegian University of Science and Technology, formerly in this department. The stream ecology team, Jason Dunn with USGS in Corvallis, Oregon, our very own Leroy Poff. And on our simulation modeling team, Aaron Landguth. Before I continue, I really feel like I need to come clean with you all and just let you know that I have a problem, which is I like frogs a lot, probably more than is healthy. All frogs are cool, but some frogs are cooler than others, and the holy grail of the frog world is the family Escapidae. Escapists are the coolest frogs in the world for more reasons I have time to talk about now. But one reason they're the coolest frogs in the world is that they are the sister clade to all extant frogs. They diverged from all the frogs 200 million years ago. So they've been evolving happily on their own for hundreds of millions of years, which is why I like to call them the seal camp of the frog world. Another reason they're the coolest frogs in the world is they live in the best region of the world, which of course is the Pacific Northwest. It literally looks like this. This is not photoshopped. And there's 2 species in this entire family. There's the coastal tailed frog, shown here in the light green, and the Rocky Mountain tailed frog, shown in purple. Escapus julii and Escapus montanus. Another reason they're the coolest frogs in the world is males have this prominent cloacal tail, which is used for internal fertilization and fast-moving streams. This is the only frog in the world that has this structure. Another reason is that the tadpoles have a sucker mouth that's used to attach to rocks where they're the most important grazers in a lot of these streams, so they play a really important ecosystem role. In fact, their suckers are so interesting that I had an undergraduate count their teeth, bless her heart, and we wrote a paper about the developmental and environmental drivers of tooth number in hundreds of frogs, which is probably why I'm still getting spam email from the American Dental Association. And yet another reason that tailed frogs are the coolest frog in the world is that they have peptides in their skin that literally friggin' kills cancer. This frog's got everything. But those are not the reasons that we decided to study this frog for this question. We're studying this frog because they live in cold mountain streams and they're thought to have really low thermal tolerance. So they should be relatively sensitive to increase in temperatures. And in fact, I wrote my NSF Graduate Research Fellowship proposal to study proto-landscape genetics of tailed frogs, but once I was in graduate school, one of my co-advisors said, oh, there's a group at UBC who's doing that, so you can't do that. And so I stopped working on them. But 20 years later, I'm working on it. So the overarching objective of this highly integrated project is to identify spatial patterns of vulnerability to climate change in tailed frogs, both within and between species, using this really integrative framework. So we collected all sorts of different types of data to get at different aspects of this, this framework that I talked about before. And the sub-question I want to focus on right now is whether there is evidence of local adaptation and sensitivity traits and or genomic data in tailed frogs. So we're focusing on this aspect of the diagram I showed you. And specifically, we're testing just simply whether there's evidence for local adaptation in these thermal sensitivity traits that has really important implications for relative vulnerability of populations at different elevations and different places on the thermal gradient. So to get this big question, our first goal was to characterize the thermal environment of these streams. And once we had done that, to test for evidence of thermal adaptation in our focal sensitivity trait, which is CT max. So once again, CT max is that maximum temperature that individuals can tolerate before they essentially lose bodily function. So here was our sampling design for this research. We had 3 different elevational gradients, both in the range of the coastal tailed frog and the Rocky Mountain tailed frog. And then in each of those areas, we had one sort of focal basin where we did more intensive sampling. And so this was led by Amanda Cecchino, this aspect of it. So we had 14 to 15 different populations in each species. And we were focused on the tadpole stage, which lives in these streams for 1 to 5 years. So that's another aspect that's really interesting about these frogs. They have a very long life history. So to characterize the thermal landscape, we installed these in-stream temperature loggers known as HOBO loggers to understanding spatial variation and temperature across streams and seasonal variation. And then to test for potential little thermal refuges in these streams, we measured at a very fine scale along these 100-meter reaches to see if there's little pockets of cold water refugia where these tadpoles can hide when it gets really hot in the summer. And then we estimated CT max. And so to estimate CT max, you first put your organisms in acclimation chamber, like in a cooler. And we actually do this with tadpoles, but we're not very good at drawing tadpoles, so I'm showing a frog. And you acclimate them in their happy temperature, which for these frogs is about 8 degrees Celsius. And then you put them in these individual containers. These were coffee filters. and you slowly ramp up the temperature, in this case at 0.3 degrees Celsius per minute. And then when you get to a point where they're no longer responsive, you've reached CT max. And then you return the tadpoles back to the acclimation chamber, and they're happily swimming along. So you're not getting to the point where they die, you're just getting to the point where their bodily function ceases. So what we found is that there's lots of variation in stream temperatures among these sites. So this shows temperature on the y-axis, and here's the elevation. In the coastal tailed frog, we saw the expected pattern of decreasing temperatures with increasing elevation. We didn't see that in the Rocky Mountain tailed frog because there's interesting things going on, like high elevation streams that are the outlet streams of lakes. So in the summer, the water stratifies, you get that warm water going into those some of those high elevation streams. So you don't see that pattern, but you see tons of variation in stream temperatures over really small spatial scales. In some cases, 10 degrees Celsius difference in temperature in less than 10 kilometers. So this sets the stage for potential local adaptation. And then, as I mentioned before, we measure that fine scale temperature variation to see if there's any of those cold water refugia. And so you're measuring temperature every half meter to meter in these streams, and it allows you to map variation in stream temperature. And this is what we expect to see in most of our streams. We have little cold water pockets where those tadpoles could hide in the height of the summer. And this is one of the streams in Oregon. Another way to view this is looking at a histogram that shows temperature relative to the average temperature in the stream. And this is what we saw in Augusta Creek. But what we saw in most of these streams was very limited variation in temperature. So there's no places for these tadpoles really to hide. So far, in terms of the environmental template, we see lots of variation of stream maximum temperatures among populations, but limited opportunity for behavioral thermoregulation. And so based on this, what we predicted was local adaptation and this thermal tolerance trait, which would be seen as a positive relationship between CT max and maximum stream temperatures. And what we actually saw was this pattern in the Rocky Mountain tailed frog, but not in the coastal tailed frog. And so we don't know for sure why there is this difference in these two species. One hypothesis is that it just gets a lot warmer in the interior Rocky Mountain tailed frogs. So they're experiencing temperatures around 20 degrees, which might require them to evolve or plastically respond with an increased CT max. But CT max is a relatively easy trait to measure, but it's been criticized as not being relevant to the temperatures that these species and these populations experience in nature. Because the CT max is way over here on the thermal performance curve, which shows how well organism performs as a function of temperature, which is oftentimes much greater than their maximum environmental temperature that they experience in nature. And so these organisms are spending most of their lives at temperatures in their happy spot, their T-opt, which is their thermal optimum. So whether CT max is relevant to that is a valid question. And what you might really want to know is whether CT max is related to thermal stress tolerance in the thermally stressful zone, which is what they're going to experience more and more with climate change. So to get at this question, Amanda set up an awesome experiment. So this was focused on Rocky Mountain tailed frogs. And what she did is she collected tadpoles from these different sites, 7 different sites, and then put them in different stream temperatures and acclimation chambers. Some of them were at their average stream temperatures, some were at their sort of maximum or near maximum stream temperatures, and some were at temperatures beyond what they normally experience now, but what they're going to experience very soon. And what she found is that there was no mortality in temperatures of 5, 10, and 15 degrees, a little bit of mortality at 20 degrees, and a lot of mortality at 25 degrees, and a lot of variation in mortality. So these temperatures that they're starting to experience are going to kill them. And so the question is whether this variation that we see in mortality has anything to do with CT max. And lo and behold, there's a nice relationship. So populations with higher CT max experience much less mortality. Small differences in CT max have huge impacts on mortality. So going from a CT max of like 29.5 to 30.5, you go from almost complete mortality to about 25% mortality. And so this suggests that maybe CT max is a reasonable index of thermal tolerance. And importantly, Amanda just put up a preprint of an epic meta-analysis that she did where she got data from all these different studies that have information on CT max and other parts of the thermal performance curve. And she found that CT max across all these species does a really good job of predicting different components of thermal performance curves, which is good news for these studies that have used CT max as an index of thermal tolerance. All right, so let's move on to the genomic results. And so our CT max results are exciting. We see variation in CT max, which might reflect local adaptation, but that could just be due to phenotypic plasticity. So we also want to get the question of whether this variation is genetically based. And to do this, we use a genomic approach. And this was led by Brenda Forrester. And this is the sampling design that she used. So she used a combination of whole genome resequencing, where you're sequencing the entire genome of multiple individuals in 4 different populations in each of the species. So 1 low elevation site, 1 high elevation site, and each of 2 elevational gradients in each species. And then she used what's called targeted capture, which is a way to just focus on looking at genotypes of a small subset of loci to expand the geographic inference of this study. So in terms of method, she had 10 individuals per site times 40 sites, so 40 individuals. She aligned the sequence reads to a chromosome level reference genome. And what she found in the coastal tailed frog, there are many loci related to CT max and or August temperatures. And also in the Rocky Mountain tailed frog, she found essentially the same results. So a large number of loci are significantly related to August temperatures, suggesting potential local adaptation related to temperature. And a lot of these are also related to CT max, indicating that this is a genetically based trait and it's highly polygenic. So anytime you show these kind of results, you have to show the mandatory Manhattan plot. And this shows the significance of a given locus, a given region of the genome, being associated with either the trait of interest in a genome-wide association study or being associated with the environmental layer of interest, in this case temperature. And what she found is, for example, this is looking at the genome-wide association study for CT max in the Rocky Mountain tailed frog. And lots of different loci are related to it. And a lot of the loci you'd expect to be related to thermal tolerance pop up, like heat shock protein modifier. Then with a targeted capture, did another 22 sites, 11 in each species. And in summary, is finding many loci that related to temperature or CT max. Some of these variants are related to the loci that make sense, like heat shock protein or loci related to hypoxia tolerance. And saw that in both the coastal tailed frog and the Rocky Mountain tailed frog. So in summary, this suggests that CT max is genetically based, it's highly polygenic, which means it can evolve. And there's also a lot of loci related to adaptation to temperature variation in both species. So what does this all mean, going back to those original hypotheses? Well, in the Rocky Mountain tailed frog, where both CT max and genomic data support thermal adaptation to temperature, this hypothesis supported. Now, it's interesting that it's not related to elevation as related to temperature, because temperature and elevation did not show that expected relationship. In the coastal-tailed frog, the hypothesis supported depends on the type of data. So the CT max results suggest that there's not much variation in CT max across elevation or across temperature, suggesting that those low elevation populations might be most vulnerable. And the genomic data does suggest that there's adaptive differences related to temperature, so potentially other thermal traits. So the type of data that you're looking at has a big influence on your inference. And so this is an example of the importance of these integrative approaches. If you just use one approach, then you're not going to get the complete story. All right, so far I've just talked about sensitivity, so this aspect of vulnerability, but we're also looking at all these other different aspects of vulnerability. So in terms of evolution as one dimension of adaptive capacity, the fact that we see that CT max is so polygenic and that we have many loci related to temperature suggests that there is evolutionary potential to evolve increased thermal tolerance. We're not done with the dispersal piece, but we have another student on a related project I'll mention in a minute who is doing neutral landscape genomics to understand patterns of gene flow and dispersal, what rates of dispersal there are, and what drives those patterns of dispersal. Amanda has also done a thorough job of looking at plasticity, in which she did acclimation experiments, keeping tadpoles either at 8 degrees or a warmer 15 degrees to see if that results in an increase in the CT max. And what she found is there was some level of plasticity, but really not much. So this value here, the acclimation response ratio, is how much CT max increases per degree increase in your acclimation temperature. And the acclimation response ratio for the tailed frogs, both species, is about half what we see in other amphibian species. So a little bit of plasticity, but not a lot. You might also be asking about the exposure component of vulnerability. So in addition to the temperature loggers we have in the stream, we're fortunate in the Northwest to have this awesome Northwest temperature database developed by USDA that's based on tens of thousands of stream temperature gauges to help us project increases in temperature. So isn't federal science amazing? So the next steps are to use these spatially explicit individual-based models to integrate all these different pieces. This is very complex, and just looking at vulnerability in a single species involves understanding sensitivity, exposure, adaptive capacity, and these different dimensions of adaptive capacity. So we're using an individual-based Eco, Evo, Devo approach developed by Aaron Languth. And what you end up in the end, when you integrate all this information, is some sort of map like this that tells you relative vulnerability across the landscape. And the goal here is not just to understand vulnerability in tailed frog populations. The goal ultimately is to understand which of these aspects of vulnerability are really most important for determining vulnerability in other systems as well. So we're really excited about these results. You know, we're almost done with what we said we were going to do in our proposal, but sometimes things happen that are not in your proposal, like unprecedented megafires. And so this happened right on top of our study area. And this was what they called a once-in-a-millennium extreme event. And one thing I think everyone is learning is that these once-in-a-millennium extreme events are no longer once-in-a-millennium. And so this landed right on top of the McKenzie River Basin, where we had our focal area. The footprint of the fire was 172,000 acres. You might have seen the previous ones, the area of Neuchâtel, which is a canton in Switzerland. I had to explain that when I gave this talk in Switzerland, how big this was. And you can see that the footprint of the fire landed on a couple of our sites, but not on these other ones, right? And we had already collected stream temperature data, thermal tolerance data, and genomic samples from all of these different sites. So we thought to ourselves, I wonder what happened at these sites. And with that maniacal gleam in her eye, Amanda and a field assistant set out to do just that. So they drove to the site, and there's all you can see is this massive fire scar, and the road ends, and then there's like a precipitous cliff. And when you look over it, it's just this gnarly slope that you have to almost rappel down. There's her field assistant. And it's really tangled mess, and actually quite dangerous because it gets really hot in a fire scar in August. But, you know, Amanda gets there and she's got a big grin on her face. And the first thing you want to do is see, like, if the temperature loggers are still there, because when you have a big fire scar, you have increasing floods, and it can wash out things like a temperature logger. She found the temperature loggers. Even before that, so here's what this looks like. Here's before the fire. It's a beautiful, magical, temperate rainforest. There's fairies running through it. All the fairies got singed. And you can see this the same spot. There's that log there, that log. There's that one with the bend. There's that one with the bend. She got the temperature loggers, and 1 year after the fire, temperatures had increased 5 degrees Celsius and the peak temperature in the middle of the summer. So we're like, that's interesting. No canopy, temperature gets hotter. And she found tap holes in this thing still, in that nuked landscape. And so she got the tap holes, brought them back to the lab, and measured CT max. And she found that CT max had gone up a ton. And so that's interesting. And it's not only going up at these sites, but these were the highest CT max values we've seen in any of our sites in this whole area. So this begs the question, A, how do they persist? How does CT max go up? Is it plasticity? Is it genetically based evolution? Is it both? And what are the impacts of this potential rapid evolution on the rest of the population, the rest of the landscape in the context of increasing temperatures? So we're thinking, well, we should write a rapid proposal. We did. This include former graduate student Lisa Shaw, another GDPE graduate, Amanda, Aaron Langoof, myself, other folks. And we're right in the midst of analyzing these data. In fact, Marty Cardis is currently doing the GWAS to see if there's been a increase in the genetic breeding values related to CT max in these sites. So Marty was a research scientist with NOAA and was part of the purge, and NOAA's loss is our gain. I also want to make the point that these extreme events are no longer rare. Leroy and I and some other people had a different rapid grant several years ago to look at the effects of the massive 2013 flood on stream insect communities and evolution. And so the lesson here is that we need to be treating all of our field sites as pre-extreme event sites and be ready to monitor the effects. Also want to just make a plug that we got a follow-up grant from a program called NSF Organismal Response to Climate Change. It is still good. And this is bringing in other species. So we're looking at the interaction between food webs and interspecific variation and sensitivity and adaptive capacity on the ultimate effects of climate change, looking at both a trout predator as well as a scapus prey. All right, so finishing up, what is the moral of the story? Well, there's 4 morals to the story. One is that local adaptation and sensitivity matters for understanding relative vulnerability of different populations to climate change. So evolution needs to be brought to the table in addition to ecology and the social sciences to ultimately understand vulnerability. Secondly, extreme events are now the norm, and we need to be prepared to quantify the organismal response to these extreme events to do a good job of improving our ability to predict the effects of climate change. Lastly, or second to last, study what you're passionate about, even if it takes 20 years to get back to it. And probably the most obvious moral of the story is that tail frogs are awesome. So I just want to finish my talk with a message of hope and empowerment, especially for the early career scientists in this room. This is kind of rough times right now. We're unprecedenting these challenges of the global pandemic, first of all, climate change, biodiversity collapse. And so where do you go to for hope? Well, I go to Lord of the Rings. And I think that this quote from J.R.R. Tolkien is an excellent message for where we're at and what we need to do. So it's really essential that we're all at the table. And there's a lot of scientists that are stepping up to the table. Here's some examples of some friends and colleagues I know who are doing this, including our very own Barry Noon, who's over in Fish, Wildlife, and Conservation Biology. And there's lots of different ways to get at the table, and CSU is a great place to learn how to do that because we have excellent training opportunities in SOJS and other programs. So start practicing this. So you can be at the table at the international level. I've been involved with various conservation genetics groups, working really hard to improve the incorporation of genetic indicators, goals, and targets, and the Convention on Biological Diversity. And thanks to this really awesome international team's efforts, the Global Biodiversity Framework now finally includes really rigorous genetic goals and targets. You can be involved at the national level. So I work a lot with people in Fish and Wildlife Service to do a better job of integrating genomics into endangered species decisions. And one of the best ways to do this is you train graduate students and postdocs who become federal scientists. And then GDPE faculty member Meena Balgopal and I are involved in writing what's called the Nature Record, and it's an assessment of the status of biodiversity in the US. And then I've got other colleagues who are working on local conservation issues, such as figuring out how to conserve boreal toads in our own backyard. So I just want to finish by acknowledging all the awesome people at CSU who make this such a great community to work in. I feel lucky that whenever I walk across campus, there's a really good chance I'm going to bump into one of my favorite people. Awesome people in biology, faculty, leadership, and staff. We have a couple of subgroups, the Fuego group and the Vlad group, GDPE. I think that has a nice sound.
Yeah.
Ecology and evolution, right? I'm sorry about the Darwinian slip. The leadership, staff, the whole community is awesome. Lots of great people over in SOJS. And just lots of awesome collaborators across campus that I've been lucky to work with. Just want to finish by thanking my partner in crime, who I've been married to for 26 years. She teaches Spanish literacy to first generation and newcomer Spanish speakers over at Poudre High School. And she has a huge impact on those students' lives. And her dedication to her students and her fighting spirit inspire me all the time. And my kids, who are now the same age as my graduate students, who always keep things interesting. All right, thanks again for inviting me to visit CSU and GDP. It's been a real honor, and I'd be happy to take any questions now or talk with you at the reception.
Time for a couple quick questions, and then we'll transition to the reception. Or not quick.
This one right here.
Oh yeah, thank you. Really interesting talk, and I might have missed it. For the two species, do you ever see hybrids? And if so, how do you think about their adaptability to the future environment?
Yeah, I mean, in a lot of systems, hybridization has pros and cons because it could add, adaptive genetic variation that helps species respond. But in this case, these species are separated by hundreds of kilometers. They have a disjunct range, so there's not really any potential for hybridization. But we can move them around and see what happens. Ed.
So just thinking about this, what's the generation time of the frogs? Is that
Oh yeah, these things are nuts. So these guys, for fish people, I say that they're basically bull trout with legs because they have the longest generation time of any frog in the world. So depending on the temperature of the stream, it takes 1 to 5 years for them to metamorphose. So they're in the stream for multiple winters. And then once they metamorphose, it can take a few to several years for them to become sexually mature. And so there's been records of them living like 30 years in nature. So it's a very long generation time, and it's an oddball in that sense for a frog.
So given the the change in the stream temperature is somewhat recent on those time scales, and would you expect genome fixed adaptation with something with such a slow?
Yes. So if there is rapid
Back
evolution,
to
yeah, if there is, what it represents is like, you have tadpoles in the stream, and a subset of them that don't have high enough thermal tolerance, they just die. And so you could have an average increase in the allele frequencies of alleles related to thermal tolerance. So it could still be rapid evolution within a generation, and that's been seen in Darwin's finches, it's been seen in guppies, it's been seen in lots of organisms. But it would represent like you're truncating the phenotypes that are there, and if they're genetically based, then you could see a change in allele frequencies and a change in breeding values related to CT max in that stream. So that's what we're testing right now.
With the fire one?
Yeah.
Thanks.
I think that, was that what you were talking about?
Yeah. Well, I was talking about the whole thing, but the fire is
Okay.
a nice example of acute.
Yeah.
I thought this was a super interesting talk, and I really enjoyed your explanation of CT max, especially for organisms that are developing underwater. But I was curious how you transfer that same measurement to like terrestrial organisms, or if you think that there's another metric that is maybe better suited for climate change for animals that develop on land rather than in this water where they can't really be.
Yeah, well, first of all, I'm going to email me and I'll give you Amanda's email address because she's the expert on this, but I'll do the best job I can. CT max is actually measured for other organisms like lizards and insects and lots of different species. So you have to measure it slightly different. That you have to determine when you lose bodily function in different ways for different species, but it is used for lots of different ectotherms in particular. But there are, there's a whole variety of different approaches you can use to measure thermal tolerance. It's certainly not the only one. Another thing that people use with like lizards is sprint speed at different temperatures to look at those performance curves. So use different temperatures and measure their performance at those temperatures. So there's a lot of different ways to do this, but it just was, frankly, the easiest way to do it in our system. And fortunately, it seems like it's ecologically relevant based on our mortality experiment and other studies.
One last one, Peter.
Thanks. Just sort of curious, that other freshwater ecologists like David Lodge are really bullish on environmental DNA sampling. I'm just curious to get your thoughts on like, if you think that that's going to give us a revolution in sort of the spatial density of genomic data, throughout these systems where you do have to sample these very small microclimates to get more expansive.
If what kind of sampling will change? I didn't catch the beginning part.
Just like, the sampling you're doing in this study of like, getting these really high resolution microclimate data, but like getting a wider sort of landscape picture through like a massive increase in sample size through environmental DNA.
Environmental DNA.
Oh, environmental DNA. Yeah. Well, I think, environmental DNA is certainly super useful for understanding occupancy of species across the landscape, and it's, exploding the field. I purposely stayed away with it because it's like a whole nother field.
Sure.
But I think, it's going to be really important for understanding changes in distributions and occupancy as a function of temperature or whatever factor you're interested in.
Thanks.
Okay, well, let's thank Chris again. Now.
What's that?
You're officially distinguished now.
Wow.
Yeah.